Hey guys, welcome to another video for Falcon Biology. In this video, we're taking a look at the Krebs cycle. We're taking a look at how glucose gets transformed, the energy in glucose gets transferred into an ATP molecule. So just to kind of go back beyond the mitochondria, we're looking at what's going on at the cytoplasm. And at the cytoplasm, glucose gets broken down, glycolysis gets broken down into pyruvate. So a 6-carbon molecule gets broken down into 2 3-carbon molecules. And if you recall in this process, 2 ATP are created. So most of the energy that was in glucose is still stored in those 2 pyruvate molecules. So we're going to follow one pyruvate molecule and take it through the entire cycle process. So pyruvate goes through a series of reactions where that 3-carbon molecule gets turned into a 2-carbon molecule called acetyl-CoA, acetylcoenzyme A. And that 2-carbon molecule, so we had 3 previously, right? So that 3rd carbon molecule gets released as carbon dioxide. And that's one of the waste products of cellular respiration, if you recall. So as we continue, acetylcoenzyme A will then group together with oxaloacetate, a 4-carbon molecule, to make a 6-carbon molecule, citrate. And that will essentially begin the process of extracting energy from those pyruvate molecules. Now, you don't have to remember succinate, malate, but I do want you to remember a citrate in the citric acid cycle because it's the first molecule that's made in the Krebs cycle. So again, as you can see the arrow going through the cycle, it goes through a series of reactions, and every step as the molecule is slowly being, or energy is slowly being extracted from this molecule to produce NADH and FADH2. So recall that NADH and FADH2 are electron carriers. These are high energy electron carriers that are going to take those electrons to the electron transport chain. And so at this point, that pretty much summarizes the first half of the cycle that we're discussing here in transforming glucose into ATP. So in the next video, we're going to look at that second half of the electron transport chain and look at it in a little bit more detail and that final process of transforming or exchanging or reprocessing, repackaging the energy that was in glucose to acetylcoenzyme A, to the citric acid cycle, to NADH and FADH2. So real quick, just to go back, what's the point of this first cycle? The point of this first cycle is to take those pyruvates that came from the cytoplasm, that came from glucose, to take those pyruvates and extract or to repackage that energy into NADH and FADH2. That's the main point of that first half. All right, let's go on to the second half in the next video.